Welcome back. We looked at in the past couple of lectures about how one can identify a lead. We looked at an overview of what medicinal chemistry is, and it's actually drug discovery is basically trying to find a lead. So, in today's lecture we look at how a drug interacts with a target. Okay. So, keep in mind this interaction is nothing but there are forces that are operating between the drug and the target. So, today we will look at some of the aspects of these. So, before we do that, if you look at a picture of a cell, the cell consists of a lipid bilayer, and typically there are receptors on the surface which is marked here, and then there are also transport proteins which I have shown here, and these are some generic representations of different kinds of proteins which we will look at in detail. And here is the carrier of the genetic information, which is DNA or RNA as the case may be. Okay. So, these are the main targets that we will look at. In today's lecture, we will look at how the drug interacts with any of these targets. Okay. So, just to recap, enzymes are the main machinery of the cell. They catalyze several reactions, and we shall look into detail about how this catalysis occurs. And receptors, as I mentioned to you, are basically the major components of signaling inside a cell. So, they are typically located on the cell surface and they respond to stimuli, and they are vital in transmission of signals. For example, neurotransmission is mediated by receptors, for example. Now, the third most important target inside a cell, nucleic acids. These molecules hold the genetic information or genetic code, and of course, without them the cell cannot function because they have the blueprint for the working of the entire cell. So, keeping these targets in mind, let us look at how drugs interact with each of these targets. Okay. So, the vast majority of drugs that are used today are targeted at proteins. Okay. So, these proteins, as I mentioned earlier, are classified as receptors which are on the cell surface, enzymes which are typically present inside the cell, and transport proteins which are in the membrane. Okay. So, it is important therefore for us to understand what protein structure is all about in order for us to understand how the drug acts on the protein. So, let us look at some of the aspects of what determines the structure of a protein. Now, proteins have 4 levels of structure. They are named in the ascending order of numbers, which is primary, secondary, tertiary and quaternary. Okay. So, the primary structure is nothing but the sequence of amino acids, and here is a box showing all the naturally occurring amino acids. We will not go into these in detail, but this is an aspect of self study that you can take up. And so, the sequence in which these amino acids are arranged is called the primary structure. Okay. And the bond that occurs within among these is known as a peptide bond. It is nothing but an amide bond, and the peptide bond is planar because of significant double bond character. So, if you see here that there is a resonance form wherein the lone pair on nitrogen goes in to the amide and forms this kind of a resonance form. And so, therefore, there is a significant double bond character between this carbon nitrogen bond. As a result, the rotation across this carbon nitrogen bond is also quite restricted. Okay. So, this is something that offers a unique character to peptide bonds, and so whatever bond rotation that happens has to happen on either side of the peptide. So, it has to happen on this side or on this side. Okay. So, let us keep that in mind when we are looking at the next to understand the primary structure. Okay. Not only there is a double bond character in the amide bond, but there are also a conformational restriction. So, here in this cis conformation there is steric hindrance which can disfavor this conformation, and so therefore, the trans conformation is more favored, and so not only the double bond plays an important role, but the conformation of this also can play a role in the way in which primary structure is present. Okay. So, the cis conformation is of course possible when you have a proline residue. The secondary structure is nothing but how there are regions where there is order that is adopted by the protein chain. Okay. So, here the main types of secondary structures are the alpha helix, the beta pleated sheet and the beta turn. Okay. So, we will look at some of these in a little bit of detail, and so here is the first secondary structure, which is the alpha helix. Okay. So, in the alpha helix, what happens is that there are intramolecular hydrogen bonds that are present, which can play an important role in the way in which the residues are arranged. Okay. And these hydrogen bonds are arranged along the axis of the helix, and it is a very important secondary structure, and it is an important component of many proteins, which is alpha helix. Okay. So, in the alpha helix, there is a coiling of the protein such that the peptide bonds make up the backbone. Okay. And there is a hydrogen bond that occurs between each of these coils. Okay. So, this is an important structure, secondary structure, that is present in a number of proteins. Okay. So, here interestingly, what happens is that the R group, which is the typically let us say it is a isoleucine or leucine or any of the other amino acids, the R group ends up being outside the alpha helix. Okay. So, this results in minimizing the steric interactions and stabilizing the structure. Okay. Now, the next secondary structure is actually the beta pleated sheet. So, here again the structure is held together by hydrogen bonds. Okay. We will look into in detail about the nature of hydrogen bonds shortly, but what happens is that this undergoes the arrangement of a beta pleated sheet is along this is something like this. Okay. Now, what happens is that the side chains are situated again at right angles from the sheet to again reduce the steric interaction, right. So, these are two important structures, the alpha helix and the beta pleated sheet, wherein the side chain is actually protruding outside. Okay. Now, a beta turn is nothing but it helps, it is another special case of intramolecular interaction, where it allows the polypeptide chain to abruptly change its direction, and as a result it becomes more globular in nature. Okay. So, here is an example of the beta turn. Here the hydrogen bonding interaction between the first and the third peptide residue occurs, which stabilizes the turn. Okay. So, we will look into in detail a little bit later about the types of proteins, but globular proteins are ones which allow the protein to have a more compact shape. Okay. And shape again is one of the important criteria of how protein function is determined. Now, after we looked at the primary structure, which is nothing but the sequence of amino acids, and the secondary structure where there are regions of order in the protein, now let us look at a tertiary structure, which is nothing but the overall three-dimensional shape of a protein. Okay. Now, as I mentioned earlier, shape is very important for determining the function, but it is also important for how a drug binds to a protein. Okay. So, in this case, so I can show you, this is a representation of a protein. Now, there are the alpha helix portions are represented here, and the arrow which is showing here is nothing but the beta sheet. Okay. Now, if you take a string and place it on a table, let us say, you will rarely find that it actually folds up itself, right. But a polypeptide does not do that. If you put a polypeptide, what happens is it starts to fold, okay, and it starts to assume these ordered structures. So, one of the ways in which we understand this folding is that because each of these amino acids which are constituents of a polypeptide have a functional group attached to it, these functional groups can interact, okay, and this interaction can actually be attractive or it can also be repulsive, and this helps the protein assume a shape which is a thermodynamic minimum. Okay. Of course, there are large number of scientists who work on this problem of protein folding, and it is something that is still a lot needs to be understood, but there is a fairly good understanding of how some of these processes occur, and that is useful for us to understand how a drug can interact with a protein. But broadly, these interactions can either be attractive or repulsive. Okay. So, what happens is that, so when you have a long polypeptide chain, typically what happens is the hydrophilic residues, which are basically the amines and the carboxylates, go out on the surface of the protein, and this is because typically the protein folding occurs in aqueous media, which is basically water, and so there is a lot of potential stabilization that occurs in water, and the hydrophobic residues, which are for example represented by this, start to go inward. Okay. So, therefore, there is a combination of attractive forces, which is hydrogen bonding on the hydrophilic residues, and repulsive forces, which is hydrophobic, and that makes the protein take to a shape where the hydrophobic interactions actually start going along together. Okay. So, this is another representation of how this long polypeptide chain can actually fold, and so here is on the left is a long polypeptide chain, and what happens is that it can actually go through favorable and unfavorable interactions, and as a combination of this it can assume this shape, and this is the tertiary structure of a protein. Okay. Now, in order for us to understand how a protein folds and how these interactions occur, we need to understand what these interactions are. Okay. And there are disulfide bonds which are actually covalent in nature. Other than disulfide bonds, all other interactions are non-covalent. But let us first look at what a disulfide bond is. A disulfide bond is nothing but a bond between two thiols, which are part of cysteines, and this is an oxidative modification where a new bond is formed between sulfur and sulfur, and this is a covalent bond. Okay. And covalent bond, the typical bond strength varies from 40 to about 110 kilocalories per mole, and it is a fairly strong bond and it is difficult to break. So, during folding, if there are two cysteine residues which are far away from each other, they can come together, and by doing an oxidative modification they can actually form a disulfide bond, which is sometimes referred to as disulfide bridge, and this helps the protein assume a particular structure. Okay. Now, these two cysteine residues can actually be quite far from each other, they do not have to be close to one another, right. So, the primary structure is therefore, there is very little information that one could get from the primary structure, which will help us understand how it can fold, because you can have two cysteine residues which are quite far from each other, which can form a disulfide bridge and push the folding in a particular direction. Okay. The second important bond that is formed is actually an ionic bond. Okay. An ionic bond is nothing but an interaction between a positively charged species and a negatively charged species. This is also sometimes referred to as salt bridge, and this occurs when in neutral pH, when you have a carboxylic acid, it usually exists as carboxylate. Okay. And if you have an amine, it exists as ammonium. So, now a positively charged ammonium species can interact favorably with a negatively charged carboxylate ion, and this forms what is known as an ionic bond or a salt bridge. It is not a very strong bond, the bond strength is somewhere between 5 to 10 kilocalories per mole, and the amino acid residues that can interact in this are aspartic acid, glutamic acid, which will exist as carboxylates in neutral pH, and the amines are lysine, arginine or histidine, which can exist as ammonium in neutral pH. Okay. The next important interaction which we referred to earlier was hydrogen bonds. Hydrogen bonds occur when you have an electronegative atom between two hydrogens. Okay. So, one of the electronegative atoms is bound covalently to hydrogen, and the other hydrogen is bound to the electronegative atom by what is known as a hydrogen bond. Okay. Now, hydrogen bonds occur typically when you have fairly electronegative atoms such as oxygen, nitrogen or fluorine, and the bond strength of these somewhere range between 1 to 7 kilocalories per mole. Okay. And if you look at the structures of amino acids, nearly all amino acids have oxygens and nitrogens in them, and so you would expect a range of these side chains to actually be involved in hydrogen bonding. But there are amino acids which have in them a side chain which can involve in hydrogen bonding, and examples of those are serine, which has a free alcohol as I showed on the right. And so, a serine can actually interact with what we referred to earlier as aspartic acid, which exists as aspartate, and it has a carboxylate ion, and this carboxylate ion can interact with serine OH very favorably and it can form a fairly strong hydrogen bond. Okay. The other residues which can involve in hydrogen bonds are tryptophan, tyrosine, asparagine, and the other basic amino acids such as lysine and arginine and histidine. Okay. The next important interaction is what is known as a van der Waals interaction. Van der Waals interactions are very weak in nature and typically weaker than a hydrogen bond, and they occur between two hydrophobic regions in a protein. Okay. So, if you look at the structure of valine, it has an isopropyl group and it can interact with another leucine which also has a similar functional group, and these can weakly interact with each other through what is known as hydrophobic interactions. Okay. And there are other amino acids which also can involve themselves in such van der Waals interactions, such as alanine, phenylalanine and proline. There are side chains such as methionine, tryptophan, threonine and tyrosine, which contain a polar functional group, but they also have some amount of hydrophobic character. Okay. So, depending on what functional group is located near it, it can either go through some hydrophobic interactions. Okay. And as I mentioned earlier, van der Waals interactions are quite weak and their bond strengths are typically less than 2 kilocalories per mole. Right. So, now let us look at the relative importance of these bonding interactions. So, when you have two cysteine residues, they can obviously form a covalent bond, and but these cysteine residues are not that often found, and there are of course many proteins which disulfide bridges, but these are a very special case. So, van der Waals interactions and hydrogen bonding interactions on the other hand are relatively more important because a larger number of amino acid derivatives can be involved in these interactions. Okay. And as I mentioned earlier, since protein exists in water, the hydrophilic amino acids will tend to be around the surface and will hydrogen bond with water, and the hydrophobic amino acids would remain buried. Okay. So, here is an example of how something can happen on the surface while the peptide chain is folding, and something that would probably end up inside the core of the protein. Okay. And it also helps explain why enzymes catalyze reactions, because some of these reactions are impossible in aqueous environment, and so these hydrophobic domains which are present in the center, they provide an important area where catalysis of small molecules can occur. Okay. So, these enzymes fold up in such a way that they usually contain a canyon hollow area which is what we call as an active site. Okay. So, imagine a spherical shaped protein, and in the cavity of this there is a cavity which contains some hydrophobic nature, wherein a non-aqueous environment is provided. And now, in this non-aqueous environment, small molecules can drift in and reactions can occur. And by small molecules, I mean fairly hydrophobic molecules, and those can go and bind into the surfaces. So, here is a pictorial representation of that. So, this is a substrate, and here is on the left is an enzyme, and as you can see, the enzyme has fairly polar nature on the top, whereas it has an area where the substrate can actually come and bind. Okay. So, there are large number of proteins which have these similar hollow areas or clefts where binding of natural ligands or drugs can occur. Okay. So, here again the importance of van der Waals interactions and hydrophobic interactions are important. Okay. Since a large number of the drugs are actually hydrophobic in nature, so these interactions will play an important role in how the protein binds to the drug. Okay. So, therefore, an understanding of these interactions is crucial to designing effective drugs that will target these binding sites. Okay. Lastly, a protein can actually also have what is known as a quaternary structure. And so, these quaternary structures are nothing but when two proteins interact with one another, and they can exist as dimers or tetramers and so on. And these types of interactions can occur, for example, when you have regions of which are hydrophobic in nature, and these can get together and they can help the protein to exist as a dimer. Okay. This provides a distinct advantage for two protein molecules because the hydrophobic areas face each other rather than be exposed to the aqueous environment. Okay. So, when proteins are synthesized, the initial code of the protein is provided by DNA, and the DNA is converted to the information is transferred to RNA, and this process is called transcription, and the process by which RNA the information in RNA is actually converted to a protein sequence is called translation. Okay. So, after the translation, the peptide sequence that is produced then folds up and produces a finally folded protein. But after translation, there can also be modifications which are called as post-translational modifications, and these post-translation modifications can assume importance in a number of disease states. We will look at some of these examples later. So, some of these proteins are modified after translation, and so for example, the N termini of many proteins are acetylated. Okay. And so, this acetylation makes these proteins more resistant to degradation by enzymes, and so therefore, it is a very useful post-translational modification to occur. And acetylation of proteins also has an important role in the control of transcription and of course, in cell proliferation and differentiation. Now, the next post-translational modification which occurs on alcohol containing amino acid residues such as serine, threonine and tyrosine. Okay. So, this phosphorylation is a very common signaling mechanism, and it is also a very important post-translational modification. And a number of signaling mechanisms are actually mediated by phosphorylation. We will look at examples of this much later in the course. So, the next type of post-translational modification is basically glycosylation, where a protein is modified with a sugar unit. Okay. And these sugars are present on the surface of cells, and they are really important when it comes to cell-cell recognition. And as a consequence, they are also key players in many diseases, and also during treatment with a drug, these help with determining the outcome of how the drug interacts with a cell.